Hello and welcome to Science Research Weekly, Season 5, Episode 10. Call me Isoptera. Get ready for fusion, plasma corrections, Alzheimer's and meat consumption, cytoplasmic tradewinds, a new termite that looks like Moby Dick, immersive dreaming and restfulness, the Artemis II Crew launch, why learning R in 2026 is a good career move, and the top 40 new CRAN packages for February 2026. This episode will cover material from March 27th to April 2nd. Let's get started. First with our research articles. First up is combined influence of rotation and scrape-off layer drifts on recycling asymmetries in Tokamak plasmas. So Tokamak chambers, they're those donut-shaped sort of fusion chambers or trying to make fusion chamber work chambers. And it turns out they leaked more than models would suggest. And it turns out actually modeling some of the actual metrics, or not metrics, but properties of the plasma itself, like drifts, rotation, viscous coupling, allows researchers to better understand how the plasma moves, why it hits some places more than others, and improve the design and operation of these chambers. All right, next up we have meat consumption and cognitive health by APOE genotype. So the APOE genotype, people with this certain genotypes, I think there's a couple different kinds, and it increases risk in dementia, Alzheimer's. And so there's a study, a cohort study that looked at what older adults without dementia and meat consumption, and they actually found a significantly improved outcomes with high meat consumption. So people with higher meat consumption had better cognitive trajectories and reduced dementia risk. So turns out high meat consumption, especially like non-processed meat, seemed to help protect somewhat against dementia, at least for this specific genotype. Next up, the landscape of microbial associations in human cancer. So been a lot of work on microbiome of cancers, and it turns out that a lot of these studies has used a single data set, the Cancer Genome Atlas, but looking at other data sets like Genomics England, which this paper did, found that only one, or actually only two cancers had associations of microbes, colorectal and oral, and only colorectal cancer had, as they would put, how would they put that? Unmistakably distinct microbial communities that could be reliably used to distinguish anatomical site. So looks like some of the hubbub about cancers having their own microbiomes is a little overbroad. That looks like, yeah, it looks like colorectal does, but most of the other ones don't look like they have a consistent one. All right, switching to cell biology here, we have the paper Compartmentalized cytoplasmic tradewinds direct soluble proteins. So it turns out proteins in cells can actually be moved around in part by cytoplasmic flow, that the cells can actually create, as the title says, somewhat analogous tradewinds to push proteins to where they need to go. So you don't really typically think about sort of flow within a cell like you would in an ocean, but looks like that is a real phenomenon. Okay, next up is avoiding and reducing microplasmic false positives from dry glove contact. So microplastic contamination is big in the news. And though it turns out that certain particles coming off of gloves can create false signals and mimic microplastics. So if you're trying to wear gloves to take samples to test the amount of microplastics in something, you actually might be adding false positive, overestimating microplastics with latex glove residue. So the authors actually recommend a nitro cleanroom glove to reduce contamination. So that's important. realization when you're trying to accurately measure something, you need to make sure you're accounting and eliminating contamination as much as you can. All right, shifting gears here, we have this next article, Cryptotermes mobydicki, Isoptera Kalotermitidae, an extraordinary new termite species from French Guiana. So, new termite species, and as the species name might suggest, Moby Dick is actually a reference to the whale from Moby Dick, the sperm whale, and it's not surprising to see why. If you look at pictures, which I have up on the video podcast, the little termite's head looks like a little tiny sperm whale. It's just great. It's not a white whale like the Moby Dick. The head's black, so I guess it's a little termite black whale. And yeah, absolutely deserving of the name Cryptotermes mobydicki. All right, we'll continue on here with the next article. Rapid mid-Cretaceous diversification of squid and cuttlefish preceded radiation into coastal niches. So looking at evolutionary history, phylogenetics, researchers found that for modern decapodiform orders, so that's squid and cuttlefish, probably originated in the deep open ocean during the mid-Cretaceous, and then after the Cretaceous Paleogene extinction event, they were able to later radiate out into things like coastal and shallow water environments where they're found today. All right, we have this article here. Immersive NREM2 dreaming preserves subjective sleep depth against declining sleep pressure. So perceived sleep depth is a determinant of subjective sleep quality. And traditionally, it was thought that unconsciousness, so the feeling of deep unconsciousness during sleep, was the main driver of perceived sleep depth. But it actually turns out something else is, which would seem slightly contradictory, but actually immersive dreaming, the perceived amount of immersive dreaming is sort of just as powerful or sort of on par with deep unconsciousness when it comes to perceived sleep depth. So if you feel like you've got a good night's sleep, you really got into a deep sleep, immersive dreaming actually might be one of the key factors rather than just feeling out of it, feeling totally unconscious, and having that reduced cortical activation. All right, final typical journal here. We have a Chelicerata-bearing arthropod reveals the Cambrian origin of chelicerates. So Chelicerata, it's an arthropod clade, includes things like spiders, scorpions, mites, and it's not entirely clear when they originated, probably sometime during the Cambrian explosion, but it hasn't been clear. But now there's this new fossil, Mega Chelicercerex castellei, which was found in the Middle Cambrian, and it has a distinctive three-segmented chelicera, so diagnostic of a Chelicerata. And so that brings that into Chelicerata certainly being present in the Cambrian, even as fairly large predatory chelicerates. All right, with that, we're done with the typical papers. Of course, I think the big science news of the week and probably of the month, possibly of the year, is the NASA's Artemis II Crew launched this week. It was really fun to watch. I actually watched with my kids, and so the 4 astronauts are now headed to go around the moon, farthest any human has gone, and that'll be a 10-day journey. So possibly next week I'll have an update on that, but so far it's looking good. All right, speaking of going into the unknown or semi-unknown, we'll go into some preprints. We've got 3 here from bioRxiv. First up is how much information is there for inferring species trees. So there's a lot of phylogenetic data sets now with a large amount of loci. More isn't always better. So the paper looks at how actually only having subsampling only the most informative loci can actually produce better species tree samples. Next article, microdiversity is higher in temperate than in virulent bacteriophages from soil environments. So bacteriophages are viruses. Turns out, looking at different baromics data sets, that the more temperate, so less deadly, less sort of infective ones, had higher what they call microdiversity. That's intraspecies genetic diversity than the more virulent ones. And final one here is backed fold, sensitive protein functional annotation across the microbial tree of life using structural information. So yeah, this backed fold is a command line software tool for ultra sensitive but taxon independent fast annotation of protein sequences across the microbial tree of life. With that, let's turn to some research tools. 3 new R bloggers posts. First from the Pharmaverse blog, Meet Data View R, the view you always wanted. It's an update to the function view called Data View R, which has different features above and beyond view, like interactive filtering, reproducible code generation, variable metadata, etc. So you can check that out if you have found view slightly lacking. Next up, why learning R is a good career move in 2026 from the Jumping Rivers blog. So going into why you might still find a lot of use out of learning R this year. From the fact that it's built for data, not just programming, the fact that it's still in high demand in analytics, research, and healthcare. It's open source. That's incredible data visualization. They say it has a beginner-friendly learning curve. You might agree or disagree, but that's the argument. And so, yeah, Jumping Rivers blog says that R is so worth learning, and I would concur with that. And finally, we have the February 2026 top new 40 CRAN packages by Joseph Rickert. So there's a lot of good ones there. Under computational methods, there is set weaver. Create sets of variables based on mutual information. Under data, there is NHANES data. Instant access to the Harmonized National Health and Nutrition Examination Survey, NHANES. Under utilities, we can get all the way down there. We have BirdDog, provides a unified set of methods to detect scientific emergence and technological trajectories in academic papers and patents by combining citation network analysis with community detection and attribute extraction, etc., etc. Finally, under visualization, there is ggInterval, extends ggplot2 to visualize symbolic interval valued data with various plots, etc. Speaking of R, closing out here, we have new CRAN packages. There's taxdiv, taxonomic diversity indices using Deng entropy. There's ggRefine, pretty ggplot2 themes, hex session, create a tile of logos for loaded packages, and insect label R, create labels for insect in collection. No new grants again, so this will be our time ended here. Hope everyone had a great Easter, and I will see you next week. As always, science on.